The phylogeny of everything. By the end of the slide presentation, you should be able to identify commonalities among prokaryotes, understand factors that differentiate archaeobacteria and eubacteria from eukaryotes, and explain strategies for rooting the tree of life. In modern biology classes, the division of life into 3 domains is accepted as fact. However, this divide has not existed for as long as one might think, and is still subject to scrutiny today. Beginning in 1925, prokaryotes were separated from eukaryotes based on what they lacked: a membrane-bound nucleus. Several scientists suggested that prokaryota was an invalid classification because it defined organisms based on what they lacked rather than their shared traits. It is now known that prokaryotes share the traits of chromosomes attached to their membranes and co-transcriptional translation. Until the 1980s, archaea and bacteria were both grouped into the kingdom of bacteria. A study on methanogens found that rRNA taken from methanogenic archaea was equally unrelated to bacteria as it was to eukaryotes. If bacteria and archaea belonged in the same kingdom, this would not be expected. The new taxon Archaeobacteria was originally formed from methanogens and their relatives. It has come to include extremophiles, like hyperthermophiles and halophiles. Archaeobacteria is distinct from eubacteria in several ways. The archaeobacterial elongation factor, ER2, contains the amino acid diphthalamide. The methionine initiator tRNA is not formulated. The aminoacyl stem of the initiator tRNA terminates with base pair U. The DNA polymerases are not inhibited by acetylcholine or butylphenyl D-GTP. And peptide synthesis is inhibited by anisomycin, but not chloramphenicol. Archaeobacteria will also differ from eubacteria in ribosomal protein composition, membrane lipid synthesis, cell wall synthesis, flagellar composition, and the enzymes involved in the synthesis of tetrahydrofolate and tetrahydromonopterin. The main factor that separates archaeobacteria from eubacteria and eukaryotes are the phospholipids in their membrane. The figure on the right side of this slide shows the basic membrane structure of eubacteria and eukaryotes, labeled A, and archaeobacteria, labeled B. Archaeobacterial membranes differ from those of eubacteria and eukaryota by containing glycerol ester phospholipids instead of glycerol aryl ester phospholipids. Their membranes have L-glycerol instead of D-glycerol, and the lipid tails of their phospholipids are isoprenoid. The isoprene side chains will have multiple branches and can be joined together. Their side branches can also form carbon rings. Archaeobacteria and eubacteria are now considered superkingdoms or domains within prokaryota. In 1990, a 3-domain system of life was established that is still used today with 3 domains of life: archaea, eukarya, and bacteria. This tree was traditionally displayed as unrooted, and attempts to root this tree have produced varying results. A method of rooting the tree was proposed where a pair of paralogous genes that exist in all organisms could be used to root the tree. For example, as shown in the figure, if gene A was duplicated into genes A1 and A2, A1 and A2 would be expected to diverge in the same order as the 3 organismic lineages. A1 sequences could then be used to root A2 sequences, and A2 sequences could be used to root A1. So far, attempts to root the tree of life with different genes have produced conflicting results. No conclusive rooted tree has been found for the 3 domains of life. In 1925, the dichotomy between prokaryotes and eukaryotes was first proposed by Chatton. From 1977 to 1980, Woese and Fox et al. Released studies on methanogens that suggested archaea should not be classified as bacteria. This was the beginning of the separation between eubacteria and archaebacteria. In 1979, Schwartz and Dayhoff suggested a pair of pyrologous genes shared among all three domains of life could be used to root the phylogenetic tree of everything. Iwabe et al. Was the first to apply this concept in 1989 using two pyrologous elongation factors, EFTU and EFG. Their study indicated that eukarya is a sister taxon of archaea to the exclusion of bacteria. Over the following years, subsequent studies using different genes produced conflicting results. Lastly, in 1990, Woese et al. Proposed a tripartite taxonomy of life, which we know is still used today.